Virology Chapter 5 Flashcards. Question number 1. A highly specific protein produced by B lymphocytes in the body in response to a foreign antigen, such as a bacterial or viral antigen, that is capable of neutralizing their effects by binding to them. Adaptive immunity. 2. Chemicals such as aluminum salts added to vaccine to improve the body's immune system response, often allowing for smaller amounts of antigens to be used in the vaccine. Adjuvant. 3. Strength at which an antibody molecule binds an epitope. Affinity. 4. A type of cytotoxicity in which natural killer cells, monocytes, macrophages, and neutrophils bind to target cells containing bound antibodies, thus killing them. Antibody-dependent cell-mediated cytotoxicity. 5. A macrophage or dendritic cell that expresses major histocompatibility complex class II, MHC2 molecules on its surface and exposes antigen peptide fragments to Th lymphocytes. Antigen-presenting cell. 6. The cellular state occurring when host cells have secreted interferons in response to a viral invasion. Interferons bind to receptors on neighboring cells, signaling other cells to produce antiviral proteins such as PKR, RNase L, and 2', 5' oligoadenylate synthetase. Antiviral state. 7. Over 30 different serum and membrane-bound glycoproteins that act in sequence when activated by a viral infection to form a membrane attack complex that ultimately causes the destruction of the invading viruses and infected cells. The 3 pathways of this system are classical, lectin, and alternative, involved in innate immunity. Complement system. Conceptual model showing interactions between the environment, the host, and an infectious agent. A change in any of the 3 parameters may favor disease conditions. Disease triangle model. 9. RNA with 2 complementary strains that triggers the RNA interference RNAi defense system. Double-stranded RNA. 10. A part of an antigen that interacts with a specific antibody often consists of 5 to 20 amino acids or monosaccharides. Epitope. 11. A rise in body temperature, a defense mechanism against some invading viruses and other microbes. Febrile response. 12. Tail region of an antibody that interacts with cell surface receptors called Fc receptors and some proteins of the complement system. It crystallizes in the cold and is a constant region of the antibody that does not bind antigen. Fragment crystallizable region. 13. Class of steroid hormones that upregulate the expression of pro-inflammatory proteins. Glucocorticoids. 14. Most common form of liver cancer commonly occurs in people with liver disease, particularly those suffering from hepatitis B or C. Hepatocellular carcinoma. 15. The premise that if the majority of the population is protected from an infectious disease through immunizations or genetic resistance, the chance of a major epidemic is highly unlikely the proportion of a population that is immune to an infectious disease. Herd immunity. 16. An abnormal deficiency of complement proteins involved in innate immunity in the blood can be a hereditary or acquired condition. hypocomplementemia. 17. Immunoglobulin M, the first immunoglobulin to respond to a viral or other microbial antigen. Serology tests are designed to detect IgM because it indicates the first exposure to a particular pathogen plays a role in adaptive immunity. IgM. 18. The class of immunological proteins that react with an antigen involved in adaptive immunity. Immunoglobulins. 19. A key feature of adaptive immunity. After responding to a pathogen for the first time, a few B and T lymphocytes remain in the body as these in the event that the body is reinfected by the same pathogen antigen. Immunological memory. 20. Protects humans against any pathogen, regardless of the species or type of infectious agent, but that does not retain any memory from prior encounters with pathogens. Interferon, phagocytes, natural killer cells, or dendritic cells. Complements and apoptosis play a role in this. Innate immunity. 21. An antiviral protein produced by body cells on exposure to viruses that trigger the synthesis of antiviral proteins. These proteins are the first line of defense against all viral infections. Plays a role in innate immunity. Interferon. 22. A cytokine produced by T lymphocytes and other white blood cells that causes other white blood cells to divide. Plays a role in adaptive immunity. Interleukin. 23. A vaccine in which whole virus particles have been inactivated. Killer vaccine. 24. Interferon induced and activated during a viral infection that destroys RNA, both cellular and viral, within a cell. Plays a role in setting up an antiviral state during an innate immune response. Latent ribonuclease. 25. A traditional vaccine that consists of a highly weakened or apathogenic virus that is capable of replication that stimulates antibody production and cell-mediated adaptive immunity. Live attenuated vaccines. 26. A set of genes that controls the expression of these proteins involved in recognizing foreign molecules. Major histocompatibility complex. 27. The first line of the body's defense in which viruses are trapped by mucus and phagocytes in the mucosal tract. Mechanical immunity. 28. The last 5 proteins in the complement cascade. C5b, C6, C7, C8, C9 self-assemble to form this. This is inserted into membranes of bacterial or viral pathogens, allowing salts and water to enter them, causing osmotic lysis of the pathogen. Membrane attack complex. 29. A cell derived from B lymphocytes or T lymphocytes that reacts rapidly upon re-exposure to an antigen. Memory cell. 30. A tool used to measure the expression of a large number of genes simultaneously or to genotype multiple regions of a genome. Microarray analysis. 31. A mechanism of autoimmune disease. Theory that similarities between a foreign, e.g. viral protein antigen, and a self-protein are sufficient enough to result in the cross-activation of autoreactive T or B lymphocytes by pathogen-derived antigens. Molecular mimicry. 32. Produced by a single clone of cells that recognizes only one antigen. These can be made in large quantities for use as therapeutic agents. monoclonal antibody. 33. A type of defensive cell that attacks and destroys virally infected cells and cancer cells. Natural killer cells. 34. This comes from receiving neutralizing antibodies from another source, e.g. receiving convalescent blood to treat Ebola virus disease patients, convalescent serum to treat hepatitis A patients, or breastfeeding an infant in which maternal antibodies and breast milk provide passive immunity. Passive immunity. 35. Conserved molecular structure shared by viruses, e.g. viral genomes, that are recognized by pattern recognition receptors, e.g. toll-like receptors that detect non-self molecules. Pathogen-associated molecular patterns. 36. These detect non-self molecules shared by viruses that are referred to as pathogen-associated molecular patterns, PAMPs, PAMPs, tagging them as foreign. Pattern recognition receptors. 37. A white blood cell capable of engulfing and destroying foreign material, such as bacteria or virally infected cells. Phagocyte. 38. Engulfment and ingestion of foreign material, such as bacteria or virally infected cells, by phagocytes. Phagocytosis. 39. Genetic variability, alternative forms within a protein or nucleic acid. Polymorphisms. 40. Pain that persists for months and sometimes years after a bout of shingles, which is caused by herpes zoster virus. Post-therapeutic neuralgia. Post-therapeutic neuralgia. 41. Fever-inducing substance, such as certain virion components of influenza A virus. Pyrogen. 42. A form of genomic immunity in fungi, plants, invertebrates, and some vertebrates that responds to invading nucleic acids. Small interfering RNAs, siRNAs, of about 21 to 25 nucleotides in length, target complementary RNAs for destruction, gene expression in a sequence-specific manner. RNA interference pathway. 43. An antiviral protein activated by 2'5'A oligomers synthesized by 2'5' oligodenylate synthetase to catalyze the degradation of both viral and host cellular mRNAs, resulting in the shutdown of viral and host protein synthesis, a cellular altruistic event. RNase-L, capital R, capital N, ACE, space, capital L. 44. The fluid portion of blood consisting of water, minerals, salts, electrolytes, proteins, such as hormones, complement, and antibodies, does not have white or red blood cells. Serum. 45. A hypersensitivity reaction by the human body after an individual is injected with anti-serum derived from non-human species that contains neutralizing antibodies toward a specific pathogen, e.g. hepatitis A virus. The non-human serum also contains foreign antigens that the human body reacts to, similar to an allergic reaction. Serum sickness. 46. A painful rash that often appears as blisters that follow a nerve. It is caused by the reactivation of the previously dormant varicella zoster virus, the same virus that causes chickenpox, during a period when the immune system is debilitated or weakened. This is most common in older adults, AIDS patients, and transplant recipients. Shingles. 47. T lymphocytes that express CD8 in association with T cell receptors, TCR, on their surfaces. They also recognize and destroy antigens that are associated with major histocompatibility complex class 1 MHC1 molecules present on virally infected cells, cell-mediated adaptive immunity. T cytotoxic lymphocytes. 48. A mercury-based compound that was used as a preservative in vaccines. Thimerosal. 49. Inoculation with a vaccine that elicits an immune response to prevent an infectious disease upon exposure to a specific pathogen. Vaccination.